let me try to start our yesterday's demo, and you maybe tell me if anything is audible. Okay. Were you able to hear anything? No? You were great. Great. So we'll be able today check at least some things that I wanted to check. That's here is the case we were not able to try yesterday. Here you see the base frequency of 200 Hz and some number of overtones. And if I start it, so we were hearing some sound, and here you see the FFT, so the Fourier transform of that sound. And here you see these sort of peaks, like overtones and the main frequency, and I can exclude the main frequency or turn it back on, and our brain is able to deduce the main frequency even in its absence. So I will start it and exclude the main frequency, and we will hear almost no change. So maybe works better with a bit higher frequencies. Let's try this. So yes, that was the yesterday's demo that we had, and I hope you were able to hear that the tone of the sound is almost the same. Well, volume a little bit goes down. That's reasonable because we have these sort of attenuation with respect to different frequencies. So that sort of exponent attenuation with these parameters. Yes. Well, maybe I can get some smaller. What about without attenuation? I didn't try, so we can check it. Oh. Well, for some reason it broke. Not so good. Okay. And here is the fixed version from yesterday's, so just some sort of reminder for you that this was the case of the system that has different parts with different resonant frequencies of these parts. And here you see that when we wobble the whole system, one of that subsystems may react to this sort of wobbling. And here was an example how that happens in our ears. So here we have that membrane, and here you see the high frequency sound, so something was vibrating somewhere here. And let me check the low frequency sound, what happens. So it should vibrate somewhere over here. Why? Okay, for some reason it doesn't want to show me the whole video. Okay, we will force it. We will force it. Okay. Close something. Good. Okay, and so today, after we have considered some sort of anatomy of the ear and how that works on some larger scale, we have more or less positioned ourselves at least inside of the head. Now we are ready to get even deeper to the next level and consider some individual cells that will be basically transforming the mechanical oscillations into some sort of electric signal, or let's say action potential, that will be fired with different neurons and sent to the brain and processed and so on. So here you see an image, enlarged image of the organ of Corti. So we have seen that yesterday in many slides. I maybe just return for a moment somewhere either here. So here you see this organ of Corti, so here it is, or you can see it right here. So here is organ of Corti. So here you see that, and this is a sort of slice. And here you can see once again some more slices, and here is that organ of Corti in this sort of slice. So we are getting into details of exactly this organ. Here it is enlarged, and it contains cells that are able to transform mechanical oscillations into some sort of signal. The auditory receptor cells, which convert this mechanical energy in the change in membrane polarization, are located in this organ, and the organ of Corti consists of hair cells, rods of Corti, and various supporting cells. So here you see this sort of tectorial membrane. So here you see the hair cells that are basically transforming mechanic oscillations into some signals that are later let's say conveyed with neurons to the brain. And here you see inner hair cells, and we will consider why do we need two types and what do they do. The auditory receptors are called hair cells because each one has 10 to 300 hairy looking stereocilia extending from its top, and these stereocilia, they basically do certain work. The hair cells are specialized epithelial cells, so they are not neurons, they don't have any axons, and in mammals they do not generate action potentials. So whatever they do, they will transform into some signal for neurons. We will get to that in a couple of slides. And hair cells between the modiolus and rods of Corti are called inner hair cells. So here you see inner hair cells, and the cells further out the rods of Corti are called outer hair cells. So here are outer hair cells. Here you see the micrograph, so whatever is happening here. So these are outer hair cells and inner hair cells. So you can see them. So inner and outer. The outer, they have this interesting shape. Good. Here are some more micrographs. Once again, the organ of QWERTY. Here you see the tectorial membrane, and the stereocilia at the tops of the hair cells extend above the reticular lamina into the endolymph, and their tips and either the gelatinous substance of the tectorial membrane, the outer hair cells, or just below the tectorial membrane for the inner hair cells. It's interesting point to notice that these cells have different parts of them in different environments, in very different environments with respect to number of different ions in that environments. And I will show you later why do they need this sort of operation method. So what else? Axons from the spiral ganglion, they enter the auditory nerve, a branch of the auditory vestibular nerve, or cranial nerve 8. So cranial nerve 8 contains some parts of the neurons from hearing, and and that's it. Good. Here we are getting a little bit closer to the mechanics of the process. So mechanics of the process is pretty interesting, I would say. So here we have different images and the micrographs on the right. What is basically happening? So here you see the tectorial membrane, here you see inner hair cells, and here you see the outer hair cells. Everything is here wobbling. So that videos I showed you at the beginning of the lecture, you might imagine how this thing wobbles in 3D. So let me maybe return once again. Where was it? Okay, let's check this one. So that's a very schematic representation, but you might imagine how this wobble influences all that structure, so all that organ of QWERTY that we have there. So this thing wobbles, and what happens to the hair cells? Hair cells, they have these sort of hairs of different height, and you see they are mechanically connected. So you see these tip link. So these tip links, they are mechanically connecting them. What's more interesting, this is connection to a potassium channel. So they can mechanically open a potassium channel. So that's the interesting part. So when they are wobbling, they will open these sort of channels, and the potassium will be able to get into the cell. To give you some idea, I have here some sort of some rope. Let me position my hand so you may see that more or less. And here you see some smaller hair cell. So this hand I will rotate it like this, and will show you some smaller hair cell. And this hand I will rotate like this, and will show you something like larger hair cell. So interesting point to notice. First of all, they have sort of direction. So when they are moving certain direction, this channel can be opened. If I move like this or like this, nothing interesting happens. If I move like this, nothing interesting happens. If I move like this, the channel gets opened, and we have some potassium influx. And you know, if we have some potassium influx, that's pretty good, because that may change some sort of membrane potential of the cell. It may change the membrane potential, and this change in the membrane potential may be the cause for certain subsequent some cascade of biochemical processes that will lead to generation of action potential with some neurons. So what do we have here? So these like very mechanistic and I guess very nice mechanism that we have. So inner and outer hair cells, they differ in number of ways, so both anatomically and functionally, and I will tell you maybe a little bit later what's something more about the difference. But anyway, so outer hair cells are cylindrical, while inner hair cells they are more pear-shaped, and the tips of the outer hair cells stereocilia are attached to the pictorial membrane, while the tips of the inner hair cell stereocilia aren't, but instead they float free in the endolymph. Most importantly, only the inner hair cells are responsible for transducing sound into the neural signals. The outer hair cells, they amplify and sharpen the response of the inner hair cells. So they are able to move that membrane and help it vibrate in resonance with the incoming sound and amplify that movement of the membrane. While the inner hair cells are basically receiving some signal. This is a pretty interesting thing actually, because you know, our ears are capable of generating sound. What's interesting, this first of all, it's pretty normal, and what's interesting, this sound is detectable. This is even used in medicine. So in case, I don't know, like for very small children, you can't ask them, do you hear something or don't you hear anything, something like that. We can use that signal from the ears. So we are giving some input, some mixture of frequencies, and we expect certain output. So we know that these hair cells will try to somehow amplify the signal, so we can take a microphone and with that microphone record some signal back from the ear, and we can measure whether at least this part of the organ of QWERTY is working correctly and is it everything okay with hearing. So that's one of the way of diagnostics for certain problems in ears. So that's pretty interesting thing, I should say. Moreover, some other animals, I don't know, like dogs, for example, they are able to generate even some stronger signals, and these signals may be even audible by humans. I guess that was the first time this was discovered. That's by hearing something strange from the dog's ear. So yes. Okay. I guess I have slide or two on that, but let me tell it you now. Good. So anyway, we are interested now, well, in getting the signal. So amplification will be later. So when this sound wave causes the stereocilia to bend back and forth, the hair cells they generate some potential. And here you see the signaling cascade that we have. So these are certain mechanical receptors. They are able to detect, they are opened when these stereocilia are moving here and there. The potassium is getting in. We get certain depolarization. This opens the voltage-gated calcium channels, so we have some influx of calcium. And now, what's interesting, I will remind you on our lecture on neurons, and I guess you should remember that influx of the calcium, that was a really big deal, because that basically started fusion of the vesicles with some neurotransmitter. So and in these cells they have vesicles with some excitatory neurotransmitter, I guess glutamate, and they fuse with the outer membrane, and activate some neuron. So here is some spiral ganglion neurite, so here it is. It will detect this glutamate and then send signal to the brain, so generate some action potential and send it to the brain, so we are hearing something. Well, and the brain will then make some sense of whatever we hear. And here is, I guess, interesting and tricky question for you. Look on this slide. So here we have reticular lamina. Here it is. Part of the cell is in the endolymph. So this part, the lower part of the cell is in the perilymph. I can remind you that they have a very, very different ionic composition. So let me find that slide. I want to just show you that composition. So here we have it. Here we have endolymph with very high potassium, so something like 150 millimolar, and just 1 millimolar of sodium. That's here. And here we have perilymph. Well, I showed you the way how it's getting to that cells in yesterday's lecture, and it has pretty low potassium and pretty high sodium. So you see, they have a very, very different ionic compositions. And now, when we are getting to this slide and we see the different parts of this cell are in the different environments, how do you think, why it's done this way? What may be the reason behind this sort of way to construct the environment for these cells? Can you make any guess? Do you remember neurons? So we had pretty similar situations there. So we were opening some channels, closing some channels, and we were generating some action potential. What happened after we have closed everything? What happened then? What should the cell do after everything happened? Like, I don't know, like neuron that has fired some action potential. Okay, let me give you some hints. So so a cell, after firing some action potential, it should somehow restore the balance of the ions on the outside of the membrane and on the inside. To restore that balance, it uses some ATP and pumps out or in whatever ions are needed. Yeah, so I hope you remember that ATP dependent pumps that were basically actively moving ions from here to there and getting certain concentrations that we need for the cell to work properly. And that process takes some time. And in here we need to react pretty quickly. So we have some, I don't know, some high frequency sounds or something like that. So some events are happening pretty quickly and we need to react on them. Do you think we have time to pump something out or in and waste a lot of time for all that job? So probably no. Then how do you think? How do these sort of structure, this sort of structure, like half of cell being in endolymph and half being in perilymph, help in this case? How can it be helpful to solve this sort of problem? Okay, I guess everyone's tired, so no ideas. Good. Then let me show you this sort of slide. Here is a little bit more information, and it contains basically all the ideas that we need. So, what the big deal with all that environment? Here we have a lot of potassium. So that's endolymph in the scalar media. So here we have it. Here we have very low potassium. So when we need to activate something, we are mechanically opening a channel here, and potassium just goes along the gradient. So we have a lot of potassium here. We have not so much potassium here. So it's getting inside the cell. You don't need to do anything. You just open it and ions flow. Now we need somehow to get back to our initial state. These channels are closed, and the other channels that are connected to the perilymph in the tunnel of Corti, they are opened. And here we have very low potassium. So the potassium from the inside of the cell starts getting out of the cell pretty quickly. That's quicker than pumping it with all that pumps. So the idea is, like, you have two very large pools, one pool with very high potassium and one pool with very low potassium. So some large pools, and one has very high potassium and one has very low potassium. And you have many other cells working to get some potassium in here and get some potassium out of here. So these are large pools. There is a lot of space and you can place many cells doing this sort of work. So they can support these sort of levels of potassium as long as we need. And our cell, be it, I don't know, some inner hair cell or whatever it is, will just be connected to this pool for a brief moment of time, will get some potassium inside, generate that sort of neurotransmitter release to activate some neurons, and then we disrupt this connection, but we create another connection here, and this potassium just goes away. The logic is pretty similar as, I don't know, like some water supply. So you can have in a house everyone to take care of its own, I don't know, its own pump and pump water from somewhere, but you know that it will take time. So you should turn it on. It will take some time to get you water then you just I don't know, make a glass of water and you turn off everything. On the other hand, you can create some large reservoir. You can pump water in there either with many pumps or with some very large pump and powerful or something like that, and then connect it with pipes to all the houses in the neighborhood, and everyone can just open the faucet and just drink a glass of water. So that's pretty simple idea that is used here. So instead of using a lot of time and a lot of energy on that sort of pumping in and out all that ions, other cells are performing that job, creating two large reservoirs with low and high potassium, and these cells are just some sort of connecting to one to another, to one to another, connects to one, activates, so depolarizes, generates some neurotransmitter release, and then connects to another, and everything goes back to its state where it was. So that's basically the logic behind operation of these cells. I will show you that in a brief moment. If anything, I just checking if anything I have missed. So here are just a little bit of biochemical details regarding that sort of stereocilia. So here you see that each stereocilium, that's just a rigid cylinder with cytoskeleton that contains some actin filaments, some proteins like fibrin, fascin, aspin. Here you see some cross-linking and color coding of that cross-linking. So here this is enlarged. Here you see the channel that will be mechanically opened and different proteins that you have in this sort of stereocilia. And here you see the base of the stereocilia that help them like move here and there. So you see they they have some sort of tapered structure, and mechanical force applied at the stereocilium tip causes the process to pivot around its basal insertion. So here you see this sort of part. So I don't think I need to get into all of that details. I hope you understand on some sort of intuitive level how that works and how these stereocilia may sway here and there. Good. Now I will show you some video, but let's recap before this video one point. So I have shown you how these cells detect some sound. So they transform some motion, some mechanical motion caused by acoustic oscillation into depolarization of their membrane and so on. And we have considered the mechanism that contains involves that two large reservoirs with high potassium and low potassium. Now can you tell me, so now not so tricky question, what will happen if liquid in this reservoir for whatever reason mixes? What do you expect? Any ideas? So if it mixes for whatever reason, what happens to the concentration of the potassium in these liquids? It will probably get the same, yeah? So you have some mixture, and of course the potassium ions, they will diffuse here and there, so the concentrations will try to get even. Then what happens to the mechanism that I have described you? So let's check it out. So it's just some, I don't know, just some general reasoning. So nothing complicated here. So I have two reservoirs, and I have some mine reservoir. Let me even draw it. I don't know. No, okay, let it be this way. And I had a high level of liquid in one reservoir, and I had a low level of liquid in the second reservoir. If I connect to the high level reservoir, I can take some water in here in my small reservoir. That's equivalent of opening mechanically that gate and some potassium ions getting to the into the inside. That depolarizes the cell. It well, releases some neurotransmitter, and some processes go as they go. Now I am disconnecting from that reservoir, and I'm connecting to another reservoir, to this one. So now I'm able to empty this thing. So I'm able to empty that and return to the initial state. Now something happens, and these two large reservoirs are, for whatever reason, they are connected. So something happened, something broke. So now I have certain level of liquid here, and I have the same level of liquid here. Am I able to perform the same thing, the same trick as I did just now? Can I fill my small reservoir with liquid from one of them and then empty it into the another one? So probably that won't work, because when even I fill in some liquid, this level will be too high for me to empty this sort of reservoir, so I'll be not able to do that. So that basically breaks the whole mechanism, and that's very bad, because if this mechanism is broken, it cannot transform mechanical energy into that neurotransmitter release, so neurons do not detect anything, and you basically don't detect any sound, and that's the problem. And this is one of the mechanisms behind the Meniere's disease. So I will show you some small video and you will see how that influences, and I hope that after my explanations you will understand why that is a problem.
Meniere's disease is characterized by tinnitus and ear fullness that is followed by severe spinning attacks that last hours to days.
Yeah, and do you hear now the sound from the video after all the tuning I have done to Zoom? Yeah, great, then you can just watch it.
It is hypothesized that Meniere's disease is due to an abnormal increase in fluid within the endolymphatic chamber found within the inner ear. Here we have the endolymphatic chamber in green and the surrounding perilymphatic chamber in blue. The vestibular membrane separates the endolymphatic from the perilymphatic chambers. The fluid within each of these chambers are different and kept in a stable equilibrium. However, for unclear reasons, patients with Meniere's disease will start to suffer from excessive fluid buildup within the endolymphatic chamber, causing the vestibular membrane to start stretching. When this happens, symptoms of ear fullness and tinnitus occur. Should enough fluid fill the endolymphatic chamber, the vestibular membrane will eventually rupture, causing the fluids between the chambers to mix, resulting in symptoms of spinning dizziness, also known as vertigo. Over hours to days, the vestibular membrane will slowly heal back together, and the fluid within the endolymphatic and perilymphatic chambers will convert back to normal, resulting in restoration of normal hearing and balance. In order to prevent fluid from building up in the endolymphatic chamber, patients are often instructed to avoid salt, caffeine, and alcohol. Diuretics may even be prescribed in order to help prevent fluid buildup. Should medical management fail, surgical intervention can be performed. With endolymphatic sac decompression surgery, the bone around the endolymphatic sac is removed and a shunt placed to physically drain fluid out of the endolymphatic sac to prevent excessive fluid buildup. In another variation of this surgery called endolymphatic duct blockage, bone is removed around the duct and a clip placed to prevent fluid that is produced by the sac from building up in the rest of the inner ear. Finally, if vertigo is still not well controlled, the nerve going to the inner ear balance system can be cut surgically. Alternatively, special drugs can be injected into the inner ear to chemically destroy the balance system.
So now you see that's pretty bad problem, and I hope you understand why that's such a disaster, even when that membrane heals and it looks like okay, but mixing that liquids destroys the balance of the potassium ions, and you just get some sort of your cells just start being non-functional. So that's no good. Okay. Here we have some more interesting images. How much time do I have? Something like half an hour. Okay. So opening the potassium channels produces depolarization of the hair cell. We have considered that. Well, whereas the opening of the potassium channels in the neurons, you might have noticed that hyperpolarizes most neurons. And the reason that hair cells respond differently from neurons is the, well, unusually high concentration in the endolymph, which yields potassium equilibrium potential to 0 millivolt compared to the equilibrium potential minus 80 millivolt in typical neurons. What's interesting, there are also neurons. In the spiral ganglion, you have something like 35 to 50,000 of neurons. And despite the fact that inner hair cells are outnumbered by the outer hair cells by factor 3 to 1, more than 95% of spiral ganglion neurons communicate with a relatively small number of inner hair cells, and less than 5% receive synaptic input from the more numerous outer hair cells. That's pretty reasonable because the one of them are basically getting you some useful signal to the brain, while the others are just for some sort of amplification, and you don't need so much information from them. Here is some small point regarding evolution. So here you see a goldfish. Yes, looks pretty strange, but that's a goldfish. And here you see the lateral line. And the organs of mammalian balance and hearing, they both evolved from the lateral line organs present in different aquatic vertebrates, including fish and some amphibians. And the lateral line organs, these are small pits or tubes along animal sides. So here you see these sort of holes. So here it is. And each pit contains a cluster of hair-like sensory cells whose cilia project into a gelatinous substance that is open to the water in which the animal swims. So here you see some enlarged scheme. So here is water coming in. So here we have some external openings. And here you see the nerve, the cupula, and the hair cells. The purpose of the lateral line organs in many animals is to sense vibrations or pressure changes in the water. In some cases, they are also sensitive to temperature or some electric fields. So maybe you have even heard more in the context of the electric fields, but the primary purpose was sensing of the vibrations of the water. And here you see once again, so these cupula and hair cells, these are the enlarged. So here they are enlarged, and if it is bent somehow, you see that they are activated and they are sending certain signals to the brain that something is happening. So some pressure has changed, or there is some vibration in the water. Okay, so that were inner hair cells, but we have some outer hair cells. And outer hair cells, they act like tiny motors that amplify the movement of the basilar membrane during low intensity sound stimuli. And this action of the outer hair cells on the basilar membrane is called the cochlear amplifier. That's what I have told you regarding that sound from the ears, from dogs and from humans and so on. Here you see the picture like this membrane, it is vibrating, and you see how it looks like if we have no outer hair cells and when we have them in place. So much larger amplitude. This is performed on the biochemical scale. That's performed with motor proteins that can change the length of the outer hair cell, and outer hair cells respond to sound with both receptor potential and change in length. So here you have a picture. Here is the tectorial membrane. So you see they are connected to that tectorial membrane. Here we have reticular lamina. Reticular lamina divides that to environments with high and low potassium. And here you have these sort of motor proteins that are contracted or expanded. And here you see a nice 3D view of one of the motor proteins. And that looks like I think I have why can't I switch slide? Here I have that may have some sort of swing analogy. So here are outer hair cells. So when it's contracted or released, so you see that it has a little bit different length, and that's pretty similar to stretching and bending the legs in phase with the swing's natural frequency. So a person on a swing can inject some mechanical energy into the system and amplify oscillations. So here you see how someone is injecting some energy into the swing, because of course it is lost due to, I don't know, some friction in the swing, some interaction with the air, so we're not living in vacuum. And of course you need to add continuously some mechanical energy for not to stop, and this mechanical energy is basically added with these sort of actions. So you are like making something longer and shorter intact with the movement of the swing. The prestin, the protein that is essential to the outer hair cells motor, is also necessary for the cochlear amplifier to function. When the gene that encodes prestin is eliminated from mice, the animals are nearly deaf. So their ears are more than 100-fold less sensitive to the sound than normal. And the amplifying effect of the outer hair cells explains how certain antibiotics, like kanamycin, damage how they lead to deafness, because these sort of antibiotics, they damage hair cells, and that's probably one of the problems, is that the outer hair cells they die. Interesting part is that it almost exclusively damages the outer hair cells, while inner hair cells, they survive. So it is thought that deafness produced by antibiotics, it's a consequence of damage to the cochlear amplifier. That of course shows us how essential that amplifier is to us. So that's So that's pretty interesting thing to notice, because you see we really need that sort of amplifier. Without it, well, the humans are nearly deaf. And there were, I guess, some cases. I'm not sure. I didn't check the literature. I guess when people got in large troubles due to excessive overuse of certain antibiotics, even on the scale of the whole state. So there were certain problems. Here are just some notions for you. Well, since we are getting into all that hearing, so here is a hearing implant or a cochlear implant. So these are devices that mainly designed to enable individuals to hear some spoken language. I will not get into all of the details how that works, but here you see how these amplify how it is inserted into the ear. So here you see the surgery procedure under the X-ray. These tiny things, they will be electrically activating appropriate neurons, and that neurons will carry certain information to the brain. So here you see how that looks in the X-ray. And of course it's not so easy, so you need to somehow take that signal from the outside and encode it so that our brain more or less understands what do we want from it. And there are some algorithms, what's interesting, developed in 1980s, let's say like multi-speak or speak, and they encode some signal, some sound into form of a spectrum, and then the spectrogram is used to activate certain of the tiny things that will electrically stimulate neurons, and they will pass some information to the brain. So here you see some electrodograms that show the voltages sent through each of the 20 electrode channels. So there are only 20 channels, and you might guess that does not restore the full capacity of the human's hearing, but at least it is able to restore some hearing of the speech. So we have pretty narrow window for speech, something like from 1000 to 2000 hertz, and that's pretty good that we are able to restore at least something, and people are able to communicate somehow. Good. So I'm not getting into that because we are scarce on time. I guess I have something like 20 minutes only. Good. Here are pathways in the brain, so how that's happening. So we have considered the biochemistry and biomechanics of the inner ear, and now it's time to pass the signal to the brain. And here you see how that goes. So through the auditory nerve, that is part of the cranial nerve 8, it gets to the cochlear nucleus. Here you see on these slides, you see the cochlear nuclei. So here they are. That's in this image. You see here the image of the brain. That's the lowest slice, this one. Then it may go either so-called ipsilateral on the same side, or contralateral on the other side. Depending on our mode of sensing, different part of the information goes ipsilateral and contralateral. Most information that we get from our eyes, that the right field goes to the left field goes to the right. So almost all information goes contralateral. Almost everything that we smell goes ipsilateral. With ears, that's something in between. I guess like 70% goes contralateral and 30% goes ipsilateral. So it's closer to 50-50. So here you see the what goes contralateral, and that's considered the main pathway. Gets to the trapezoid body, then to the superior olivary complex. I guess we have seen that. Then it goes in the midbrain, it goes to the inferior colliculus. So there are these sort of four colliculi, and information gets here. Then from here, part of the information goes to the medial geniculate body in each side of the brain, and some part goes contralateral to the other side of the brain. And then they go to the auditory cortex. And the auditory cortex, so here you see that on this map of the brain, that's the last slice. Here it is, and here this area is even shown on the brain, on the side view of the brain. That's the primary auditory cortex, where you get the information from hearing, and then this information is used to build up our representation of the surrounding environment. So we have both ascending pathways from ear to the brain, so that we transmit information to the brain to make sense of it, and we have some descending pathways that go from the brain to the ear. They are basically carrying signals between the auditory cortex, subcortical auditory structures, and the ears. So like inhibitory neural signals from the superior olivary complex to the outer hair cells, they cause a reduction in the motile response of the outer hair cells. So you are like controlling that amplifier. They are thought to protect ears with that acoustic reflex. So you remember the tiny muscles that were attached to ossicles in the midbrain, so they are controlled with the descending pathway. And of course, the descending signals are also involved in attention. They block task irrelevant ascending auditory signals while passing task relevant. So you know that if we hear a lot of mixture of sounds, we can somehow discern which sound we're interested in, and just ignore all the rest. So here is interesting. One more interesting thing. That's a sort of coincidence detector. So here you see the superior olive. Here you see different neurons, and these neurons, they get some inputs from the left and from the right cochlear nuclei. So do I have it? So here is just a part of it shown, but you see that superior olivary complex on the left gets input from these from the right cochlear nucleus, and superior olivary complex on the right gets input from the cochlear nucleus. And you might guess that if we draw another cochlear nucleus, it will give some input here and it will give some input here. So they get inputs from both cochlear nuclei. So only half of scheme is shown, but by symmetry you might guess what happens. And here you see different neurons taking inputs from them, both. And depending on the length of the axon, it might happen that some inputs will coincide at certain neurons, and that neurons will activate. So they are detecting the coincidence. And depending on which neuron activated, we know some time difference between signal coming from the right ear and from the left ear. And that's pretty cool because we can use this information for, let's say, different purposes. One of them is basically maybe, let me check. Yes, I guess I will show you. One of them is basically detecting the source of the sound. So when the sound is positioned somehow with respect to our head, one of the methods to detect where this source is basically calculating the difference, the interaural difference between the signal coming from the source to one ear and to the other ear. Let me return. Let me return back. So here we have these sort of detectors. I guess I will show you them a little bit later. What else? The auditory cortex. So here is the auditory cortex, and here you see how different neurons are tuned to different frequencies. So there is still some frequency relation in these cortex even despite its pretty high level of the sound processing. So here you see that there are neurons, some are tuned pretty narrowly. So here you see characteristic of narrowly tuned neuron. Some may be broadly tuned. So here are characteristics of the broadly tuned neuron. What's more interesting, if you draw these sort of characteristic with respect to different frequencies, you see there are a few different parts. Each of them takes the whole spectrum of the sounds, and they are attached to one another in a pretty peculiar way. So here you see one part, that's A1. It starts with high frequencies and ends with low frequencies. Then you have a rostral core, starts with low frequencies, ends with high frequencies. And here you have rostrotemporal core that starts with high and ends with low. Now, how do you think why they are attached this way? Why don't they all start with high and end with low? Or start with low and end with high? Any ideas? What could happen if I hear some, I don't know, some low sound? I will have some neurons activated. Let me take this color in here. So they will probably somehow influence the neighboring neurons. So that's biology. It's not like some precise thing. So if something is activated here, so it will probably somehow affect its neighbors. And if we had, let's say, this rostral core starting on this side from the high frequency neurons, it will probably somehow activate some high frequency neurons and you will get some sort of mess. But if they are passed to one another with the low frequency end, even if something activates here, that's pretty okay. Anyway, you are hearing low frequency sound and something should have been activated here. So that's absolutely okay. So I hope you see the reason. If we turn them around, that may be a little bit of a problem. So some low frequency sounds will cause some areas activate, like in case of high frequency sounds, and we will get this sort of map. Okay. So here you see in that auditory complex different parts, like parts that goes what and where. So one of them is more involved in the processing what is the object emitting that sound, and the other one is involved in the processes where is the object emitting that sound. Here you have this sort of brain that shows different pathways, like these what and where pathways. The interesting thing is that we have connection between what we see and what we hear. So all these pathways, they come together, and we are basically processing the visual and the auditory stimuli together to create some image of the world around us. And now I guess, since we are so scarce in time, but I want to show you some interesting things. Here is an illusion called McGurk effect. Let's make an experiment this way. So I will run this video, and what you do, you close part of your screen. Let's say the left part of your screen you close with the hand. So like you see two copies of the same man here on the left and on the right. So please close one of that copies, just putting your hand there, look on the other one, and then write me a comment. What sound did you hear? So what he was saying. Okay, ready? I guess yeah. Let's go.
Ba 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 ba.
Okay, and write me in the comments what kind of sound do you hear.
Ba 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 ba.
B or F. So ba or fa. Yeah.
Ba 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 ba.
Okay, and now I will close the other part of the screen.
Ba 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 ba.
Music, yeah.
Ba.
Okay, and now you have closed the left part of the screen. Now close the right part of the screen, and we will try the same thing. Okay.
Ba 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 ba.
So depending on which phase you close, you should hear a different sound. So in one case it's ba, in other case it's something like wa or fa or something like that. There is one more interesting illusion. I will run right now. I will run some video. You will hear that people are, I don't know, singing something, and you will see a text on the screen. If you think that whatever they say matches that text, if you think that whatever they said at that moment matches that text, you count it, let's say, as 1. You will see a few excerpts of the text during the screen time, and you count that. If all matches, you will count some number of matches. If one has matched, you count that. And then you give me in the comments the number of times that the text matched whatever they were singing. Good? Ready? Let's go.
This crowd chanting.
That is embarrassing! That is embarrassing! That is embarrassing! That is embarrassing!
Your prime.
Okay, so how many did you count? One. Okay. 0. Good. And what did you hear? Okay, anyone else counted? No, nobody else counted. Okay, good. Do you know what they were actually thinking? basically they were thinking that is embarrassing. So I don't know, that's some soccer match, and anyone from their team or from the opponent's team did something bad, I guess, and they are now, they don't like, they think it's embarrassing, and they are now thinking that is embarrassing. And the thing is that once again we are connecting some visual cues to the auditory information that we have, and with this sort of information we are able to generate our view of the external world and get some guesses of what is happening. So the thing is that this phrase was the same all over the course of the video, but in many cases people are hearing exactly what is written, despite that they are thinking something pretty different. Well, maybe except some football fans from England that are often in these sort of situations, so they probably know that by default. Okay, I guess we are a little bit out of time, so maybe I will not get into all the rest what we have here. These are some more slides. If you're curious, you can check them. These are slides regarding such interesting thing like phase locking. So many sounds, they have frequencies much higher than the frequency our cells are able to react, and our cells are using so-called phase locking, like firing action potentials during certain phase of the signal. And if you have many cells that fire during signal during certain phase, you get this sort of volley effect or volley principle. So here you see soldiers, they were firing one after another, and you get some volley of all that shots. And here you have the same thing with neurons that are firing one after another and get you this sort of information. One more thing, I guess we had no time, that's the localization of the sound. Here you see different methods of localization. First of all, that's acoustic shadow, because our head is shadowing basically the source of the sound, and we can somehow estimate where the object is. You can play around with my demo. There is some attenuation on the left and on the right ear, and probably you will hear the difference in position between the sound source. Of course, some interoral difference that you get from the when sound comes to different ears, and difference in phase can be used. That's the method as well. The other interesting thing, we are good with localizing sound in the horizontal plane, taking into account all that acoustic shadow and so on, but we are not so good when localizing it in the vertical direction. And here the pinna comes into play. So it somehow distorts the sound depending on the frequency. Different frequencies are differently distorted, and our brain, well, recognizing somehow that sound and knowing what should it be and how it comes distorted from the pinna, is able to calculate the vertical position of the sound source. In nature, there are certain birds like owls, for example. They need to catch their prey, and they need this sort of 3D orientation, and to handle the problem they use asymmetrically set ear openings. So here you see ear openings in the owl, and you see that they are asymmetric. These are not ears, that's just feathers. Ears are hidden inside, and you see that they are asymmetric. Of course, they have some feathers, some special that are specialized to somehow distort the sound and focus that into their ears to help them orient. we are pretty bad when perceiving distance because there is not enough information in the sound. Only if we recognize the source of the sound and we know more or less how loud should it be, we will be able to estimate the distance. But you know, that's pretty okay. And I fully discard the vestibular apparatus. There is a pretty similar mechanism. The only thing that these sort of stereocilia, they are wobbled not by the acoustic sounds, but with the liquid in these sort of channels. So here you see them, and here they are positioned. So here you see these channels. Here you see the direction of the depolarization. That what I have said you previously regarding that cilia. That they have some preferred direction, and the liquid inside, it has some inertia. So if we are moving our head by inertia, that liquid is trying to stay in place, that affects the appropriate organs and activates activate that cells. So here you see how that looks like. So we are reacting to the gravity. You see the utricle and the cells, they are reacting when I'm moving the head. Here you see what happens when we wobble the head to the left and to the right. They are giving these sort of different number of spikes, and you see that this movement activates some of the cells depending on their direction. So remember that thing with rope I have shown you. So here the rope has pulled open the channels. You have many spikes. Here the rope is not pulling anything. Here you see that sort of mechanism that detects the inertia of the liquid. So here it is. So when we are rotating clockwise or counterclockwise, you see what's happening. And this cupula that is pretty similar to what I have shown you in fishes. In fish, it moves, some cells are activated and send signals to the brain. And here you see this mechanism that contains these sort of three semicircular canals. These are these canals positioned in the human's head. They are not horizontal, they are a little bit like 30 degrees turned up, but that's because when we are walking, we are looking down, so not to fall somewhere, I don't know, in an uncovered manhole, for example, and then they will be more or less horizontal. So if we are looking down, they will be positioned horizontal. And here is the MRI. Here you can see, or that's computer tomography, maybe computer tomography. So here you see some slice that shows you these parts, so this canal, and here you see these black things that are, I guess, these canals, so this and this. And here is an example what happens when we are rotating our head, and that liquid by inertia moves the stereocilia and activates different neurons. Once again, you see that thing in a slice. That's a microphotography. Here you see the crista ampullaris. So here is the ampulla, and this thing basically wobbles as we move our head and lets us detect where are we moving. Here you see once again the pathway to the brain and whatever is happening here. So I'm not focusing on that. Maybe I will show you. There is interesting connection between how do we move our head and our eyes, because we need to somehow look at some object, and there is an interesting reflex. Maybe I will show you just this video.
The third ocular motor control system is going to be that of the ocular motor reflex. I want you to look at that point right at the very distant part of the wall. Keep focused right there and don't move your eyes. Keep focused, and I'm going to turn your head quickly, but you keep looking over there.
So that's
up
not
and down.
conscious movement. That's not conscious movement. That's just a reflex. What's maybe more interesting, if you, I don't know, some turn around a few times so that you feel like dizzy and everything moves around, rotates around you, and if you, I don't know, make a video of your eyes, you will see that your eyes are moving left to right, left to right, left to right. So that's because you have all that liquid swirling inside the head. The brain thinks that we are constantly rotating, and it tries to compensate for the eyes positions, and because the eyes are moving, you see that everything around you is moving, is rotating around. So yes, good. I guess with that being said, we are complete with our lecture 14. If you have any comments, questions, please fire away. Okay, no comments, I see no questions, everyone's tired. Good, then have a nice evening and see you next week.